So we will be discussing about brain-eating amoeba. What this infection is, there has been a lot of cases recently in Kerala about this infection, and around 80 cases have been reported with 21 deaths have occurred. So we will be looking at what this organism is actually and what kind of infection it causes, what is the method by which it spreads, and what can we do to prevent the infection and how it is treated. So all this will be discussed in this lecture today. So what is this infection which has caused so many cases recently in Kerala? Why we are calling it as brain-eating amoeba? Does it really eat the brain? And is it safe to swim in fresh water these days? We'll be getting the answers to all these questions in the coming slides. So first of all, what is the organism causing the disease? Basically, the disease is caused by a free-living amoeba. What is a free-living amoeba? These are one-celled organisms that are freely found in the environment, usually warm waters and soil. So there are many free-living amoeba that are known to cause human diseases, but these are very rare infections like acanthamoeba, balamutia, naegleria, and sapenia. All these organisms can cause infections in humans but rarely. So what is happening in Kerala is this brain eating amoeba is Naegeria fowleri, which is causing infections, and basically this infection is acquired from soil or warm fresh water. So in these cases mostly it is swimming in the fresh waters like lakes or ponds, rivers, and very rarely in poorly maintained swimming pools also. Why it is called as a brain eating amoeba? Basically it doesn't eat the brain, but it infects the brain tissue and destroys the brain tissue. Since the brain tissue is getting destroyed, it has been given a name as a brain eating amoeba. And the issue is that though the infection is very rare, but the fatality, or you say the mortality, the death of patients is very high. In earlier cases, it has been seen that 95 to 97% of the infected patients have died. So mortality or the death of the patients is very high and alarming. Fortunately, in case of Kerala, the mortality is quite low, that is around 24%, and it can be probably due to early case detection and better treatment in these cases. So water containing the amoeba, how it infects or goes into the human being. Water containing amoeba goes up to the nose. When somebody is swimming with their head down in the water, then this amoeba can goes through the nose. There is a cribriform plate which makes a barrier between the nose and the brain. It can cross that cribriform plate and then goes into the brain and infect the tissue. So this infection is called as primary amoebic meningoencephalitis. Meningo means affects the meninges, that is the covering of the brain. Encephalitis means affecting the tissues of the brain. So this is primary amoebic meningoencephalitis. So let's look at the signs and symptoms of this infection. So the classic symptoms are the usual symptoms that happen with any meningitis case, that includes headache, fever, nausea, and vomiting. So these are the usual and most common symptoms that will happen. Now in fatal cases, unfortunately, it may lead to coma and death after 5 days. Other symptoms that are common to meningitis will may be present in these cases also, like neck stiffness, confusion, loss of balance, and hallucinations. Now let's look at the risk factors causing this infection. The most important and most common risk factor is diving or swimming into the fresh waters, ponds, lakes, or poorly maintained swimming pools. Now if somebody is diving or swimming into a swimming pool or a freshwater lake that is having these amoeba, then these amoeba can enter into the nasal passage of the diver or the swimmer, and from there it can move upwards along the olfactory nerves through the cribriform plate into the brain and can cause the infection. So this is the most common mode of infection. Another reason could be using tap water for rinsing the nasal passages. Now if people are using this tap water to rinse the nose and this tap water has these amoeba, brain eating amoeba, then it can enter into the nasal passage and into the brain. Now very rarely some recreational water sources also not having enough chlorine can be a source of infection. Now it must be noted that this infection is not caused by swallowing or drinking a water which is infected with the amoeba. Also, this infection is not caused from others, that is, it cannot be transmitted from one person to another person. If one person who is infected is coming in contact with another person, then this will not be transmitted, because we have seen how the mode of transmission happens. It happens through the nasal passage. If the water enters the nasal passage, then passes through the olfactory nerves and cribriform plate, then only it goes into the brain. So these are the major risk factors which should be avoided to avoid the infection. So how can people protect themselves from getting this infection? One option is using nose clips. When diving in any unknown swimming pool or freshwater lakes, it is ideal that they use a nose clip to prevent moving of water into the nasal passages. Also, keeping the head above the water surface so that water doesn't enter the nose can be another way to avoid this kind of infection. And don't dig or don't dive into shallow waters. Also, using some distilled water or boiled water or filtered water for rinsing the nasal passages is recommended. Also, since there is already a lot of cases happening in Kerala, so it is better that these people or the people around this area avoid diving in any unknown lakes or ponds for swimming in these cases. Now we'll look how this infection is diagnosed in a laboratory. The most easy method is microscopic examination. Either the cerebrospinal fluid or the brain tissue can be used for observing under microscope. So the trophozoites are quite motile and observed in the CSF. In this way, we can diagnose or identify Naegleria fowleri. PCR or polymerase chain reaction is a molecular method, genetic method, and it is the gold standard. The only issue with this is that it is not easily available everywhere and it has a very restricted availability. Then there is next generation sequencing. Of course, the availability is an issue. Here the whole genome will be sequenced and the organism is identified. It is also a good method, but the issue with this method is again the availability. It's not available everywhere. Also, it is a costly investigation. Now let's look at the treatment options available. Combination treatment is usually used to treat these infections like amphotericin B. Amphotericin is an antifungal antibiotic but can be used for treating Naegleria fowleri. Then miltefosine, again it's an antiparasitic but it is again used for treatment of Naegleria, and rifampin. Rifampin you might have heard in treatment of tuberculosis. So this combination treatment is often used. Then other options are azoles like fluconazole. This family of antifungal antibiotics are also used, and supportive care is of utmost importance to maintain the patients. Thank you for listening and let me know your feedback about this lecture.